Good morning, good afternoon, or good evening to wherever you're located in the world. Welcome to our webinar and thank you for joining us. Today, we will focus on the quality requirements of peptide-based in vitro diagnostics. A few words about myself before we start. My name is Stefanie Dubitz and I'm Group Leader Custom Synthesis at Bachem. Prior joining Bachem, I did a PhD at ETH Zurich, which focused on peptide-based tumor targeting ligands. I joined Bachem in 2017, and I'm responsible for our European and Asian customers, supporting them with peptides for in vitro diagnostics, cosmetics, and R&D. Before we start off, I would like to say a few words about Bachem. Bachem was founded in 1971. And when you calculate, you realize that we have celebrated our 50th anniversary this year. Ever since our foundation, Bachem has been focused on high quality peptide manufacturing. And very recently, we also added oligonucleotides to our portfolio. Furthermore, we are listed on the Swiss Stock Exchange. We do have 3 focus areas, which are research and specialties, CMC development, as well as commercialized APIs. Now, within each of those focus areas, we provide you products from very early stage research all the way to commercial peptide and oligonucleotide APIs. Alongside those products, we also offer comprehensive services, for example, analytical method development and validation or regulatory support. We do have 6 sites worldwide. Starting off on the left-hand side, we have 2 CGMP certified sites in California, namely in Torrance and Vista. We have 1 ISO 13485 certified site in St. Helens in the UK, which is also the site that today's webinar will focus on, and our Center of Excellence for Custom Synthesis Peptides. We have 2 further GMP sites in Switzerland, namely in Bubendorf and Viona. Bubendorf is also where our global headquarters is and where I am located. Last but not least, we also have a sales office in Japan for more direct support of our Asian customers. Now I would like to switch gears a little bit and also introduce you to in vitro diagnostics before we start off. By starting, what is actually an in vitro diagnostic or an IVD? This is any kind of medical device that's used in vitro diagnostic testing. This can be a reagent, a product, or any kind of instrument. Now, in vitro diagnostic tests themselves are performed with samples that are taken from the human body. They're indispensable for making correct diagnosis and therapeutic decisions. Furthermore, they can provide prognosis and predict treatment response. Why are peptides important in IVDs? They play an important role and can be classed into 2 categories. The first category are peptide biomarkers. Peptide biomarkers, or the degradation products, are degrading or increasing in a fashion that can be used to detect either the onset or the progression of the disease. And one example are reliable predictors for Alzheimer's disease. For example, there are concentration changes of amyloid beta in CSF or blood already a lot earlier than Alzheimer's disease actually breaks out. Furthermore, also tau proteins change in the CSF and blood long before the disease breaks out. And both of these changes can be detected in immunoassays. The second part of peptides are peptide epitopes, which correspond to parts of pathogens. These peptide epitopes are then recognized by antibodies that are produced in the human body. And this is actually where the diagnosis takes place. So the antibodies in the human body can then be detected, for example, via an ELISA, by using those peptide epitopes as the diagnostic peptides. One such example is the detection of latent infection with Mycobacterium tuberculosis, which is detected via interferon gamma. Interferon gamma is released by human immune cells after incubation with Mycobacterium tuberculosis, peptide epitopes. Now, why should we use synthetic peptides for these in vitro diagnostics? If you manufacture peptides chemically, you can also add chemical modifications to those peptides which are not present in nature. This can be helpful, for example, in order to attach them to specific surfaces onto your in vitro diagnostic. Furthermore, they can be produced with high purity and a controlled purity profile and in a highly reproducible manner. With this, we would actually be at the end of our introduction into in vitro diagnostics. And to get to know you, our audience tonight, a little bit better, we would like to ask you our first poll question. This is, for which IVD do you plan to use synthetic peptides? Is it A, for infectious diseases, B, for other diseases, C, for both, or D, are you maybe not planning on using synthetic peptides in the future? Thank you all for voting. And we see that most of you will use IVDs for both infectious diseases and other diseases. With this, we would like to move on from our poll question and have something more technical for you, which is housekeeping. You will be unable to use your camera or microphone, but feel free to write us any kind of questions that arise with you during the webinar into the Q&A section down here at the bottom. And we will have time to answer those questions at the end of the presentation. With this, I would like to introduce to you our expert speakers of today, which are my colleagues, Julie Marley and Joanne Capener. Julie Marley is our site manager at Bachem UK, and she did her PhD at the University of Manchester. At the same time, she was a peptide chemist at Bachem. Prior joining Bachem, Julie did a one-year stay at the R&D group for peptides at AstraZeneca. She held various positions within Bachem and is our site manager at the UK site since 2013. Joanne has studied engineering and has 15 years of experience in leading life science companies that have produced IVDs and Class 2 medical devices. She is our QMS manager since last year, and she is a trained lead auditor for ISO 13485-2016. And with this, I hope you enjoy the webinar of my colleagues Julie and Joanne. And I leave the floor to you, Joanne, and I'm looking forward to the Q&A session.
Thank you, Steffi. Hello, everyone. As Steffi says, I'm the QMS manager here at Bachem UK, and I'd like to welcome you all this afternoon. I'm going to speak a little bit about the regulatory side and the associated standards. So in vitro diagnostic devices, as Steffi rightly says, are used to make medical decisions. In fact, around 70% of medical decisions are based on IVD results, even though IVDs only account for around 2% of healthcare spending. Now, there was a recent change from the IVD directive to the IVD regulation in 2017. The reason for this being that the IVD directive is around 20 years old. And with the growth in innovation and new technologies, it was very clear that it was starting to fall short of current market requirements. So this led to the switch to the IVD regulation. And as a result of this, the diagnostic industry is now facing stricter hurdles for product approval. So what is the difference between a directive and a regulation? So a directive can be thought of as a goal where compliance sits in the hands of the manufacturer. A regulation, on the other hand, is a binding legislative act which forces manufacturers into compliance. Now, with the old IVD directive, around 80 to 90% of products were self-certified by the manufacturer as conforming, and only 10 to 20% required an independent notified body assessment. As you can imagine, this did lead to some safety issues and to a lot of concerns about certain products that were being placed on the market. So with the new IVD regulation, we've basically switched places. So now 80 to 90% of products placed on the market do require an independent notified body assessment, with only 10 to 20% being self-certified. So what's changed recently? If we look at the In Vitro Diagnostic Directive, this was a set of general rules that each EU member state would transfer into their own national laws. But this all changed with the IVD regulation. So with the IVD regulation, the requirements are directly applicable now in all EU member states. There is now no room for local interpretation of the requirements. And there's a lot of requirements that have changed. So we were familiar with requirements for post-market surveillance, for example. But we also now, depending upon the classification of the device, have additional requirements for technical files and labeling, which includes information in many various languages. The IVD directive had no requirement for businesses to employ risk management other than for software. But the IVD regulation now contained an obligation to establish a system for risk management. And even though not explicitly cited, the risk management is in line with ISO 14971, which is the risk management standard. There are also some new requirements as well. So, for example, post-market performance follow-up is a new requirement. And this requires proactive collection and evaluation of clinical data for risk management. And this ensures continuing suitability and performance of the devices on the market. Another new requirement is for a performance evaluation report. This is a planned and systematic collection of device data, which is used to verify the safety and the performance and also the clinical benefits of that device. So what is a standard? So a standard is basically the best way of doing something. And for those of you not familiar with ISO, ISO are the International Organization for Standardization. And when we think ISO, we think quality, we think trust, and we think credibility. Now, there are two forms of complying with the standard. So you have accreditation and certification. So accreditation is where a business is aware of the guidelines that ISO set out to define certification. Whereas certification, this is where a business is independently inspected to confirm compliance to the standard. And we believe this is where the weight sits. Now, the IVDR regulation is applicable in Europe. However, in the USA, the mandatory quality system regulation for medical device distribution there is the 21st Code of Federal Regulations, Part 820. And this is commonly referred to as 21 CFR 820. Now, the FDA worked in collaboration with ISO to revise the latest version of ISO 13485. And the result has given us a quality management system that not only covers the requirements of the IVDR and the medical device regulation, but also covers the requirements of 21 CFR 820. So ISO 13485 is now globally accepted standard that meets all the requirements that we need for our regulatory bodies. Now, lastly, I wanted to talk a little bit about a standard that most people are familiar with, ISO 9001, and why Bachem opted for 13485 instead of 9001. So with 9001, these are both quality management system standards, but we believe with 9001, there are a lot of gaps there. I'm going to talk a little bit about these. First of all, risk management. As I've mentioned, 13485 has that mandated requirement for risk management activities. 9001 promotes risk-based thinking, and this is used for 9001 to minimize customer dissatisfaction. With 13485, we're using risk management for medical devices to ensure that those devices placed on the market are safe and effective. Key processes as well. So 9001, it allows each business to determine which procedures they would like to document. Whereas 13485 mandates key procedures must be documented. It has requirements for document control and also for control of records. Supplier control, so 9001, it talks about interested parties, anybody that your business is working with to provide products or services. But 13485, the focus is on supplier evaluation and selection and approval of those suppliers, and going forward then, constant monitoring of those suppliers. Continual improvement is a tool that's used in both 9001 and 13485. But again, with 9001, it's a very generic tool that's used mainly to enhance customer satisfaction. But with 13485, this is a tool that's invaluable for maintaining the adequacy and the suitability of the quality management system that is being used for medical devices. Then there's the authorized representative. So with 9001, a business is left to determine which responsibilities it would like to define. But with 13485, there is a requirement that an authorized representative is appointed who is responsible for that quality management system. And lastly, we have the medical device file. Now, I spoke earlier about the new requirements for technical documentation. So your medical device file is the complete history of your product, and it's invaluable to any medical device manufacturer. And this is why we believe that ISO 13485 is the perfect fit for IVD manufacturers. And with that, I'd like to pass on to my colleague, Julie, who's going to talk a little bit about the technical side.
Thank you very much, Joanne, and also to Steffi for the very nice introduction to in vitro diagnostics and into the quality and regulatory requirements. So I'd like to continue the webinar, and I will focus more on the technical aspects of peptides used in vitro diagnostics. So, as Steffi mentioned earlier, my role is a site manager for the Bachem UK site, which is based in the northwest of the UK in St Helens. And this site is a centre of excellence for the manufacture of research chemicals. We manufacture a wide range of custom and catalogue products. We use a synthetic method, and we use solid phase peptide synthesis. And we have an expertise in manufacturing a wide range of peptide modifications and across a wide scale, so from milligram to multigram quantities. We can efficiently produce over 1,000 peptides annually, and we have a range of automated state-of-the-art equipment for the upstream and downstream process to allow us to do that. More importantly for this webinar, the Bachem UK site is ISO 13485 certified, and it specialises in the manufacture of peptides for use as critical raw materials for IVD application. And in addition to supplying the peptides as materials, we also have a full range of associated services that we can offer. So a little bit more about the Bachem UK journey towards ISO 13485 certification. Joanne has explained the reason why this certification is so important. And we should emphasize that quality has always been a strong commitment for Bachem and is so for the Bachem UK side. But we decided to support our IVD customers further, that we would take a business decision to target ISO 13485 certification. After some many years of hard work, in 2017, we achieved the ISO 13485 certification. The scope is for the manufacture of synthetic peptides used as raw materials in medical devices. And at that time, the version that we were certified for was the 2003 version. Now, there were some changes to the standard that required us to be able to very quickly transition only a year later to the 2016 version, which we did successfully. We then have post-2018 annual surveillance audits, which is a requirement of the certification. And then every 3 years, as part of the cycle, we completed a recertification audit. And in 2021, we were recertified. And the site now has ISO 13485 certification, which is valid for the next 3 years until 2024. So with Bachem UK, you have a fully certified ISO 13485 site that gives you an absolute guarantee of quality. As Joanne mentioned earlier, we feel that the weighting is very much towards certification and gives you an advantage over our suppliers that might only have accreditation. So in addition to the ISO 13485 certification, Bachem also has developed a range of product grades, which are suited to our customers and their requirements. We offer a Bachem standard research grade, which usually meets the needs of most of our customers. The key elements to this research grade is an absolute guarantee of quality in the material. We also have generation of batch production records. So every batch that we manufacture will have an associated record that contains all the information related to the manufacturing processes. The final product will be analysed in quality control and released according to specifications, and the product will be delivered with an analytical data sheet that ensures that we are compliant to the specifications. And the grade and the service is flexible to customer requirements. So for example, we can dispense materials in quantities according to a specific customer demand or in very specific customer defined vials. We realise, however, that this Bachem standard research grade may not be suitable for our IVD customers, and we developed a new grade, Bachem Research Grade Plus. The Research Grade Plus offers all of the key elements of the Bachem Research Grade, but it has additional elements which are designed specifically to meet IVD customer needs. The guarantee for Bachem Research Grade is the ISO 13485 certification, the regular audits that we described, and the 3-year cycle of recertification, and we can provide our certificate to customers on demand. The additional elements of the Bachem Research Grade are critical change control. This is a key element of the standard and a requirement for all medical device manufacturers. Bachem UK has change controlled within its quality management system. We have a standard operating procedure that defines the Bachem UK approach to change control. And it helps our customers guarantee that when working with Bachem UK, that they have full control of change control. We can also offer, on customer request, generation of a master production record. And a master production record is product specific, and it contains all the information for the optimized methods to manufacture a product. And it's used to make sure that we have reproducibility and minimal batch to batch variation. The master production record is then used as product specific record to generate a batch production record, which is both product and batch specific. We can also offer our customers enhanced traceability, which will help us in investigations where we look at root cause analysis, maybe for non-conformances. And with enhanced traceability, we can include in the master production record or the batch production record specific information related to the critical chemicals, solvents, reagents, and buffers that are used in the manufacturing process. In addition to the Bachem Research Grade Plus, and again to support our IVD customers, we have an a la carte menu of Research Grade Plus services. So these services include audit support, generation and support of medical device file, generation of peptide mixtures, and stability testing. So I'll go into a little bit more detail in the services that we offer later into the presentation. So we have our ISO 13485 certification. We have our product grades which we've developed at Bachem. And together that allows us to be a perfect partner to support our customers from early stages, R&D, through to commercialization. If we look in a little bit more detail at each stage, at the very early stages where we have a customer who makes an inquiry, they will have a dedicated sales representative. They will provide us with the peptide sequence, the product specifications, and the analytical requirements. And we will manufacture that material with a Bachem research grade. If the project progresses further through to process development, many of our customers will request us to manufacture independent lots. If they take a validation approach, it's usually 3 lots that we would generate. Bachem can support with manufacture of independent lots. These can also be manufactured using research grade to minimize the cost at this point in the project. And it allows Bachem to develop the optimized processes for upstream and downstream manufacturing. When the process is adequately developed, we can then start to define the specifications. Usually our customers will generate a product raw material specification sheet that contains all of the product specific specifications. Bachem will take that customer document and they will transfer the information into a Bachem specification document, and that ensures that we do not miss any of our very important customer specifications in our documentation. When the process has been developed at this stage, we can generate a master production record, as I mentioned previously, and we will, as always, for our final product, be able to generate and deliver an analytical data sheet. And the final stage towards commercialization. We are very happy to work with customers if they want to set up agreements with Bachem. So that might be an agreement to include quality requirements. It may be around supply, or it may be around technical, or all 3 aspects. And we're more than happy to work with our customers if that's a preference. However, if they decide to place a purchase order and provide us with the defined specifications, we can also work via this route. Again, at the commercial end, we will always produce a batch production record and an analytical data sheet for the finished product. So in summary, as a leading partner in IVD, Bachem can support you through each step towards commercialization. I mentioned about the a la carte menu of Research Grade Plus services that we have available, and I'd like to go into a little bit more detail with each one now at this point in the presentation, and I'll start with audit support. Manufacturers of medical devices can undergo a number of triggers that would prompt a notified body audit. That might be related to medical device reporting, it might be product recall, it may be due to recertification or annual surveillance, or to unannounced audits. The very good news is that Bachem can provide support for all of your audit requirements. We have an extensive team of internal auditors with many years of experience. And as we mentioned previously, our QMS manager, Joanne, is ISO 13485 lead auditor qualified. It's maybe important to mention at this point that whilst the FDA have been conducting unannounced audits for several years, the EU Commission requirement for notified bodies is relatively new, and IVD products will be subjected to unannounced audits every 5 years. So we expect the number of unannounced audits to increase quite significantly in the future, and we're more than happy to support our customers with that increased requirement. As Joanne mentioned, we would expect to have regular supplier control audits from our customers. As a supplier of critical raw material, that would be an expectation that would happen. Usually it's every 3 years, but that's dependent upon the customer cycle for supplier control, and we're more than happy to support. We usually would have on-site audits, but given the recent coronavirus pandemic, most businesses have needed to adapt, and I'm very pleased to say that Bachem has adapted accordingly, and we've successfully completed a number of remote audits, and we can hold either remote or on-site audits, very much depends on the customer preference. And as we mentioned, we should consider unannounced audits, and we're very happy to support our customers with any unannounced audits, either that we can provide them information that's needed on a platform that can contain secure documents, so we can very quickly respond to any of the unannounced audits at the customer site, or we can host unannounced audit at our site if it's related to a critical raw material that Bachem UK has manufactured. Next, I'd like to talk about medical devices, and Joanne's already referenced this in her introduction. So the ISO 13485 requirement for medical devices, it states that the organisation shall establish and maintain for each medical device type or a medical device family, one or more files. They should contain or reference all of the documents that are generated that are specific to that medical device, and they should demonstrate the conformity to the requirement of this international standard and compliance with applicable regulatory requirements. Now, it's important to mention that Bachem UK doesn't manufacture medical devices. Therefore, there is no requirement for us to create a medical device file. However, as we do manufacture critical raw materials for use in medical devices, we've made a decision that we will generate a medical device file for all of the critical raw materials that we manufacture. So the contents of this medical device file would include, but not be limited to, any specification document. So we've mentioned earlier, it's either customer specification document or the transferred Bachem specification document. Any questionnaires, so these are usually related to the initial quality questionnaires that Bachem UK would complete in response to supplier evaluation, selection and re-evaluation. They would contain all the information from any audit reports, either unannounced or regular supplier audits. They would contain all the information for the customer agreements, and we mentioned quality, technical or supply agreements. All the batch production records or the master production records, which are product specific or product and batch specific. And finally, the analytical data sheets for all of the raw materials that we manufacture that would contain the quality control analytics and demonstrate conformance to the customer product specifications. Another service we offer is support with stability testing. We realise that manufacturers of medical devices have a consideration to determine the shelf life of the medical device and therefore to set an expiry date. We can guarantee at Bachem that we deliver the peptide at a very high purity at the point of QC release. But it's very important to note to our customers that peptides can be sensitive to environmental conditions such as temperature and pH, and any changes of those conditions over time and across various storage conditions. So we can determine the peptide by chemical analysis. It's usually done by HPLC, high performance liquid chromatography. That gives the guarantee of purity at the point of QC release, but it also allows us to perform chemical analysis after storage and generate stability data for the peptides, which we can provide to our customers and help them or support them to determine shelf life for their medical devices. And Bachem can support you with your customized study. So we can change the storage conditions that you would like us to do the chemical analysis on. It's very dependent upon customer requirements. We will provide you with a full report at the end of the stability study that would help you set the shelf life and expiry date for your medical device. I'd like to show just an example of the chemical analysis we do. So this is the HPLC test for peptides. This is the original QC release data. What you can see that will be generated during stability report, is that over time and under various storage conditions that the peptide can undergo degradation, various degradation pathways, and impurities can be generated. And the measurement of those impurities and the report that we can provide will help you with your setting of expiry date for your medical device. So finally, I'd like to talk a little bit more about peptide mixtures. So Bachem has a wide range of experience and expertise in generating peptide mixtures. So many medical devices will only contain a single peptide. But however, to increase the specificity and the sensitivity of a medical device, Our customers will often use a mixture of peptides, sometimes scanning a protein sequence. And peptide mixtures come with very specific challenges. They cannot be analysed the same way as individual peptides to determine purity or identity. So we have to come up with a very different approach to generate them. I'll show a visual in a few moments that show the approach that Bachem UK takes, but we have a robust process, full documentation to generate efficiently peptide mixtures according to our customer demands. So we start off with the individual peptides, and the example that we have here is to generate a mixture of 5 peptides. We have the bulk peptides that are released to high purity on QC release. We then would go through an aliquoting process so that we generate the same number of aliquots as required for the final bottles that are delivered to the customer. In this example, 4. The next process is that we would consolidate the peptides aliquots into sets, and then we would mix the peptides into the bottles, which are the final deliverable. And we will provide to the customer, according to their specifications, a heterogeneous mixture of peptides. Now, the example I've given here is for 5 peptides and 4 bottles, but it's important to note that we can deliver any number of peptides and any number of bottles. It's very, very dependent upon what the customer requirements are. So with that slide, we get to the end of my presentation. I'd like to thank you all for your attention. I hope that we've been able to show that 13485 is a perfect fit for peptide in vitro diagnostics, and that Bachem is a perfect fit for you as a supplier for your critical raw materials for your medical devices. So I'd now like to hand back over to Steffi, who will do another poll question and then move to the Q&A session.
Thank you so much, Joanne and Julie, both for your expert insights. And now that we've talked a lot about requirements, we would actually like to ask our second poll question to our audience, so to you today. Which are the most important requirements for you? Is it full traceability of critical raw materials, critical change control, no process changes or batch-to-batch reproducibility, or are all of the points mentioned above important to you? And as one would expect, most of you have answered that all of the points above mentioned are important. And this is also what we at Bachem see, and this is why we wanted to talk about all of these points together with you and mention them. Now with this, unfortunately, we are at the end of our webinar today, what Julie quickly mentioned already. In case you would like to see some of the important points that we mentioned a bit more summarized, we do have a brochure on in vitro diagnostic peptides, which is available from our website. So please feel free to download this one. Of course, the webinar will also be available on YouTube afterwards. I hope that you really enjoyed this insightful talk from my colleagues, Julie and Joanne. And now it's time for questions. And I will quickly move into the Q&A chat, and we will start off with the first one. So Julie, Joanne, be prepared. The first question from the chat is, if ISO 9001 and ISO 13485 are different, should I certify for both? Joanne, I think this is one for you.
Okay, so Bachem again faced this decision. Obviously, they are both quality management system standards. But when you're a manufacturer of an IVD, I would certainly always go for 13485. Many years ago, the two standards were very similar. In fact, 13485 was based on 9001. A lot of people refer to it as 9001 plus. However, since the latest revisions, they have diverged quite significantly. So for IVD manufacturers, no, don't certify to both. It could be quite time consuming trying to meet the requirements of both. I think 13485 is the one to choose.
Thank you so much, Joanne. And we will move on to the second question, which is, how does Bachem address the potential issue of cross-contamination for peptides? Julie, I think this would be one for you.
Yeah, thank you, Steffi. That's probably one for me. So maybe it's worth to mention that within our quality management system, we have procedures that define the cleaning processes, and they are extensive. And so therefore we can guarantee that we would expect with those procedures in place and applied that we wouldn't have any cross-contamination. But we did actually perform process validation protocols, and that took every step of the manufacturing process and looked for cross-contamination and confirmed that we didn't have any cross-contamination. So that's an additional step that we performed to guarantee that cross-contamination. And we also have another option. If any one of our customers decide that based upon their risk analysis, they would prefer to completely eliminate any risk, then they can request that they purchase dedicated materials. So that's columns, glassware, etc. And we can use the dedicated materials either for a single peptide or for a mixture or a group of peptides. And all of those measures together will manage, I think, the customer expectation for removal of any peptidic cross-contamination.
Thank you so much for your feedback, Julie. And I think here's another one for you. For the generation of peptide mixtures, can you expand on the methods you would use for dispensing and mixing?
Yes, absolutely. So I didn't go into a huge amount of detail. We had the visual there, obviously. We would aliquot peptides as powders, and the process that we can use is either manual, and that is something that we do apply for a relatively small number, but we also have a fully automated powder dispensing system, and we can use that to very quickly, efficiently, and with full traceability produce aliquots for any number of peptides. So there's a carousel that will allow up to 30 aliquots to be dispensed within a few minutes. So we'd apply either manual or automated processes. And then the consolidation process, the mixing of peptides. For the moment, that's a fully manual process. We will hope for the future to be able to move to an automated process, but it's still a very efficient process. You say we have to make sure that we have all the documents completed and the records to show that we've done the aliquots and consolidation according to the standard requirements. So for the moment, that's a fully manual process.
Thank you so much. And I think here's one more for Joanne. Does an unannounced audit count as a surveillance audit?
No, unfortunately not. It would be a very good world if it did, but the scope of an unannounced audit is typically very different to that of a surveillance or a recertification audit. A recertification audit or a surveillance audit covers the full scope of the quality management system. But for unannounced audits, it's a very focused scope and it deals with just the product. So no, it would be very nice if it did, but unfortunately not.
Thank you so much, Joanne. And here's the next question. This is, can you provide a certificate of analysis instead of an analytical data sheet or ADS? Julie, I would suggest this is for you.
It is, and I think the only difference between the two is the pagination. So Bachem has made a decision that we would generate analytical data sheets and not a C of A's. There is, apart from that signature, there is very little difference between the two. We generate our analytical data sheets from a global system. So they're automatically generated, they're linked directly with the ERP system, and it allows us to very efficiently produce ADSs with very low risk for errors against peptide sequences or data, etc. So no, we wouldn't, but I think the ADS would be more than sufficient for a customer requirement.
Thank you. And the next one is, would your peptides for IVDs being offered as non-commercial lab developed tests, for example, under CLIA, be produced to the same quality standard as those for IVDs under commercial development? Julie, I would say this is for you as well.
Possibly. I don't have a huge amount of experience for that specific application, so I'll be very honest and say I would imagine that the answer is yes, they would be the same. I couldn't see where there would be a difference, but quite honestly, I don't have a great deal of experience around the CLIA application.
And then we have one final question, which is again on peptide mixtures. If a peptide mixture is delivered as a heterogeneous mixture, how does the customer then homogenize?
That's probably for me again,
Yes.
Steffi, isn't it? So yes, the answer is we have to deliver heterogeneous mixture of peptides. That's because we use a powder dispensing and a dry powder mixing, but the customer can make that mixture homogeneous. In order to do that, they have to find a common solvent. Now, that's not always very easy to do. It depends upon the range of peptides in the mixture, and it depends upon the characteristic of the peptides. And so the answer would be to find a common solvent. Bachem can support, and for every product that we manufacture on our ADS, we do actually do a solubility test, and we provide solubility at least at 1 mg per mil in a biocompatible solvent. So if we can use that solubility data, it may be that can be used to determine a common solvent, and then the heterogeneous mixture provided to the customer can then be dissolved to provide a homogeneous solution, and then that can be used directly by the customer. So it would be important that the customer ordered the bottles in the quantities that they would like to use immediately for sample preparation.
Okay. Thank you so much for this answer. I do not see any further questions in the chat, so we would stop the Q&A session here. In case there are any additional questions or thoughts you would like to share with Bachem, please feel free to write us an email to webinar@bachem.com or contact us through any of our social media channels. Again, thank you so much for joining this webinar. Thank you for being part and making this a lively Q&A session. Thank you, Julie and Joanne, for sharing your expert insights. And we would look forward to see you at our next webinar. Thank you and goodbye.